Okay, good afternoon, everyone. As Dr. Ellenberger introduced, my name is Malia Callier. I had that wonderful opportunity of being in the Davis lab this summer. And here I just want to unpack the question of, does GBA influence the spread of Lewy pathology via extracellular vesicles? Sorry about that, technical difficulties. To start, I would just like to take a moment to really just unpack how Parkinson's disease is really affecting so many of our aging population. So it is the leading motor disease and the second most common neurodegenerative disease. It affects 1 to 2% of individuals over the age of 65 as of now. While there are clinical characteristics that many people are aware of, such as stiffness and slowness of movement, there's also other non-motor cognitive symptoms, such as cognitive decline. Now, the breakthrough neuropathology of the disease is the presence of aggregated proteins known as Lewy bodies. They contain ubiquitinated proteins, alpha-synuclein oligomers, and lipid membranes, which you can kind of see pictured in dark purple at the top. Now, for Parkinson's disease in particular, the spread is characterized by a very particular temporal spread, starting in the brainstem before moving up further into the brain, which you can see along the bottom. This spread is correlated with the rate of clinical progression. As of now, mutations in the GBA gene are the strongest risk factor for Parkinson's disease. This is significant because having the mutated gene increases your chances of developing the disease by 5 times. And as you can see in the bottom right over here, we've also found that having the mutated disease increases the rate of progression for those with Parkinson's compared to those who do not have the mutated version of the gene over the same period of time. So it not only increases your chances of having it, but it also increases your rate of progression. To better comprehend how GBA affects the progression of Parkinson's, the Davis lab utilizes both Drosophila fruit fly models, which have relatively high genetic similarity to humans regarding GBA, and iPSC-derived neuronal cell culture. Now, you may be wondering, why are we examining extracellular vesicles, or known as EVs? Well, extracellular vesicles are very, very small bilipid membrane vesicles, which contain proteins, lipids, and nucleic acids. They're very important for cellular communication within the body. As you can see in the top, we have some dark blue ones. When they're entering the cell, they are known as endosomes, and in the dark red, when they're leaving the cell, they're known as exosomes. And we believe that the GBA mutation may be affecting the clinical progression of Parkinson's by impairing the autophagy of the EVs. This is the body's way of getting rid of unnecessary factors or parts of the cell in the proteasome and lysosome. And we believe it is leading to increased amounts of ubiquitinated proteins and accelerating the Lewy body progression. Now, you may be wondering, how are the EVs analyzed? Well, first, we have the iPSCs-derived neurons, which are grown from the fibroblasts given from patients, and they're grown in the lab. The supername that they're in is then extracted and filtered multiple times. They're also sorted by size using size exclusion columns. One is shown here that I got to use in the lab. They're then visualized using a nanoparticle analyzer known as the Zeta viewer to just mostly confirm their identity before being put on a Western blot in order to further examine the contents that are inside. From my own EVs that I was able to filter and study, we have found using an anti-ubiquitin antibody, as you can see on the top of the Western blot, that they do indeed contain proteins that are ubiquitinated, meaning they were meant to be taken either to the proteasome or lysosome. But about the bottom with the CD81 antibody, we're able to basically confirm that these are EVs, seeing as this is an anti-EV marker. In parallel, the Drosophila fly models are bred using different drivers, which determine which genes and mutations will be expressed by the offspring, so we can measure the differences between them. Right now, one major project we're using the flies for is to see if the oligomerized alpha-synuclein protein in particular from the thorax travels to the brain and whether or not this is mediated by those extracellular vesicles. On this, Anna Park, who is in the lab, has found that the flies from the GBA mutation and have a muscle driver are found to have high molecular weight alpha-synuclein in the head compared to wild-type controls, as seen by this Western, which is taken from the heads of the flies. On the left, you can see that those expressing the control, so they do not have the GBA mutation, but express the GBA mutated gene in the thorax, did not have the same aggregates compared to those with the GBA mutation. This is significant because it suggests that the alpha-synuclein in the thorax are traveling via EVs to the head to form alpha-synuclein aggregates, which could indicate the start of Parkinson's progression. So overall, based on the findings that GBA promotes the spread of alpha-synuclein in flies and that the EVs can be successfully extracted from the iPSCs-derived neurons with its contents examined, our current hypothesis is that GBA mutations lead to excessive EV release and increased pathogenic protein aggregation in recipient cells. In the future, we would like to further study how the presence of alpha-synuclein aggregates in the iPSCs-derived neurons and in the flies. We would also like the opportunity to isolate these EVs and put them on new neurons to see if that also causes an aggregation. I'd like to thank Dr. and Mrs. Ellenbogen, along with Dr. McDonald's, for helping make this possible. I'd like to thank the Davis Lab and anyone in for giving this opportunity, and I'd like to thank you all for listening today. Thank you.
We have the Davis Lab up here.